Welcome to Revolution Against Evolution. I'm your host, Doug Sharp, and I have with me Peter Borger, who is an author. His book is called Darwin Revisited, and it's How to Understand Biology in the 21st Century. And I think one of the things that I would like to talk to Peter about is the fact that molecular biology, as it is really understood by most people in the media in the world and throughout the classrooms, as it is taught, is a lot different than what we actually are finding in the laboratory today. And Peter is a molecular biologist, and I am interested in seeing his perspective of how you came to the conclusion that what is understood in molecular biology is a lot more conducive to the creation point of view than it is the evolutionary point of view. Welcome to our show, Peter.
Thank you very much that you also have me here in the United States, where I happen to be for a few days, so thank you very much.
So where do you live right now, Peter? Are you in, I understand you're in Switzerland, is that true?
Yeah, it is like this. I am Dutch originally. I traveled quite a lot over the world. I lived in Sydney, in Australia for a while. I worked there. And then I traveled together with my wife back to Europe because we had our son in Sydney, and we wanted to be closer together with the family, and we ended up in Germany on the border of Switzerland where I worked.
Okay. So you're working in Switzerland, and you work now as a molecular biologist?
Yes, indeed.
And are you working in private industry now, or in the?
I worked for the university.
Okay.
In Basel and in Zurich, and I'm now a freelance scientist, in fact.
Oh, that's good. It's a little bit easier to be open with your creationist point of view
exactly.
when you're.
Sometimes it's quite hard.
I remember my experiences in chemistry and in biochemistry in college, where the creation point of view was more of a curiosity than it was. People were not as openly hostile back in the 1960s because it was so brand new. But I remember the books about that A.E. Wilder Smith wrote, and I think he's a triple PhD in the area of genetics and with biochemistry, and those things were important to me when I was going to college, because it was I was one of those guys who tried to create life in the test tube, to make amino acids come together to form proteins, and I came to an impasse. And though my professors couldn't answer my questions, I found that there was some hostility for me to come to the conclusions that I had. People would like the idea that life could assemble itself, and
Exactly.
so you have some very unique perspectives on this, and you're bringing me up to date on some of the new research. Can you tell me a little bit about that?
Yeah, of course I can. Well, it is like this. I did PhD studies in the Netherlands, and in those days it was still an enigma, in fact, how the cells of higher organisms were regulating their gene expression levels. So what they did, in fact, in those days, they started to make mice where they knocked out certain genes to get an idea about the function of these genes in the offspring. So if you knock out the gene, there should be a kind of a phenotypic effect, how the mice behave, how do they have diseases. And a very intriguing phenomenon then observed was that a lot of these mice did not have an effect at all. So you could knock out a gene in these animals, and it was as good as its brothers and sisters, which still had this gene active.
So that is really unique. Now, what do you think that really means then?
Well, this could mean several things. For instance, it could mean that there is an enormous backup in your genome, or there is a network, in fact, that these proteins work together with other proteins, and that certain proteins have the similar or the same function as other proteins because they simply can be bypassed in this network. So if you get out one of these proteins in the network, the function is immediately, in fact, taken over by another protein. So this tells us that natural selection does not really act on this type of proteins. Because if you can knock out the protein and it has not an effect, then what would be the driving force of natural selection to select this type of proteins? So proteins that operate in this type of networks cannot be selected, in fact. This is what I came to realize.
Well, that's amazing. One of the things I remember talking to, when I was studying this in microbiology, and they were talking about how there was a different dimer of two of the different nucleotides, thymine and thymine. If they're next to each other in the genetic code, the ultraviolet light might bind them together and then make that part of the gene non-functional. Well, there apparently is a repair mechanism that will excise that out and put them back the way they're supposed to be. And so it's almost as if you have your you bought a truck with that feature where if you run into a light pole, it would repair itself. And,
yeah, exactly.
And that's something that you wouldn't expect from something that came about through just natural selection.
Process, yeah, of course. Now, this is what we see in biology. We see on all levels, we see that the status quo has to be maintained. We see this also in populations or in genes that mutate, for instance. Natural selection is always there to maintain the status quo, and it's not going to evolve organisms in a certain direction. No, it is the other way around. It keeps the status quo of the organism. So there are really forces in the organism that try to keep the organism as it is, within a certain limit, of course, because there is adaptation and there is also, of course, variability within the organisms.
But the term for that is stasis.
Stasis, yeah.
And so,
What
would you tell a student to look for in his studies to give the true picture of what actually happens in the cell, in the biology?
Yeah, okay. Well, the thing is, I think what we learned, and I think this is also a very important message for students, but also for people in general, is that biology is not based in matter. It's not a matter of matter, but it is a matter of information. What we see in the cells is all coded information, or the result of coded information. The DNA we know now is coded information, and there are so many codes in this molecule that is beyond belief almost. Because we always talk about the protein code, but this is just a code to make tools. But to make these tools, you need regulatory sequences. You need switches to switch on the tools when they are needed in different cell types. So we see that all sorts of codes and codes upon codes are programmed in the DNA or in the proteins that are associated with the DNA. For instance, the histone proteins. They are also regulated, and they regulate the expression patterns of all these tools that are made and that are required for doing the biochemics in your cell. So it is all based on information, and it is not based on matter, as people still teach our students.
Yeah, histones are extremely complicated, aren't they?
They are big proteins, and the interesting part is that when you look at it, there is no evolution at all in the histone proteins.
Oh yeah.
They are completely the same in all organisms. And why is this? Because the protein needs this one function that it is keeping the DNA together, and it has specific sequences that also are able to that are involved in another code that we call the histone code, and the histone code is required also for expression of your proteins and of your genes.
So what brought about the writing of your book? You have a lot of the conclusions that you've come to in your book. Could you tell us a little bit about that?
Yes, one of my conclusions is first, and I think this is not my only conclusion, but I think that is the conclusion of most people nowadays that work in biology, that it is based on information, and that information is something not material, and information comes from an immaterial source. So it tells us that biology is from a source that is over matter, so an intelligent source or an unphysical source. This is what one of the conclusions. The other conclusion is that natural selection is unable to generate information, biological information. Another conclusion, which is very clear from the data that we have nowadays, is that the universal common descent, as the Darwinians or the evolutionists want to have it, is not existing. There is now evidence that there is, which is called polyphily, which means that there is more than one origin. This is absolutely proven biological science. So this common descent, universal common descent, that we come from bacteria, is simply not so. This is one of the conclusions.
Well, that's amazing, and I remember years ago there was a professor, I think at Baylor University, and his name is Michael Pollini, who wrote an article about that life is more than physics and chemistry, and I think that was one of the first articles that I saw as a student that really got my mind thinking about the role that God plays in the development and His creation of life, and so I really at that point began to really believe that creation, as it's taught in the Bible, was actually the best way to explain how life got here. Now, when was your first revelation to you that this is indeed the case?
Yeah, exactly. This was in fact a sort of turning point in my life, because I have been struggling my entire life with the existentialistic question: where do we come from? And by studying biology, I got more and more the feeling that this was a designed concept, that a cell is a designed concept. And the more you look into it, the more it becomes clear that it is a designed concept. And after, I think, 10 years of study, I realized that I had to write this down for myself first. And in fact, this was around 2000, 2001 that I had this feeling that there is much more into biology than just this material thing that we had been taught in the academia. So then I started to write this down, and this was in the days that I was in Sydney, in fact. And my wife was already a Christian, and well, we discussed it, and every now and then I went with her to the church, and I always had a bit of I was also a bit hesitant all the time, but when I started to realize and talked about it, I found out that it had very good cards. And then, as I said, in Sydney we went to a very modern church with a lot of music. It is called Hillsong Church.
Okay.
And that's in fact where I was touched by the Holy Spirit, by the Lord, and from about 2002, 2003 this change appeared. And since then I'm in the faith and in Christ, and I was baptized in Sydney in 2003 as a Christian, together with my wife. So it was a very happy moment for us.
No, it certainly has its benefits to adopt the Christian faith and to allow the Holy Spirit to work in your life and to control you. It's taking the biology that we see where God controls it and apply it to by choice in your life. Now, is there a difference between your view of this and what was once called vitalism?
Yeah, vitalism is more or less, as I understand it, that there is a spark in the living, in the living, that is not really defined where the spark comes from. I think this spark of vitalism comes from the designer that designed everything and who revealed himself in the Bible, in my opinion.
I remember also talking with Kevin Anderson, who's the director at the Creation Research Society, Van Denler Research Center in Arizona, and he talked about the DNA code and the code within that manufactures everything and keeps everything alive in the body as being a fourth dimensional computer program, where the information is not only, a typical computer program is one dimensional. You have a sentence of things that the instructions that follow, but all these instructions are contained in a three-dimensional model that changes according to time, and to hear him describe what happens. I remember also learning about antibodies and how they actually on demand for transporting foreign objects out of your body will conform to the specifications of that object and attach itself to it and then transport out of the body. I just was amazed by that.
This is also a very amazing design, because you have millions and millions of antibody for every intruder. You have a potential solution. And if you think about it, if this is all in the genome, it would be so big, it could not be contained in one cell. So what has the cell present? It has a system, a genetic system, where it is able to generate these antibodies in such a way that it can compress all the DNA. So it has in fact a genetic variability system. And this is very intriguing, because by compressing all these data, you can put it in the nucleus. And if you look into, for instance, the biology, where we see variability and where we see adaptations happening and where we see speciation, because remember that if you think about Noah's Ark, there were not so many animals present.
Right, yeah.
So there must be a kind of an biological system that is able to generate in a very fast way novel adaptations, novel speciations. And I have in my book described also that it is comparable to how this enormous amount of different antibodies can be generated within a few weeks. So you can also imagine that within a few years or a few hundred years, you have the complete Earth covered with species. And this is in fact what we can see in the genome still, the remnants of it.
Is this epigenetics?
This has also to do with epigenetics, but it has also to do with so-called transposons. And this is these little very little genetic elements that can hop around in your genome, but it can also very fast modulate your gene expression and gene expression patterns, but it can also swap chromosomes. So you get different patterning of your chromosomes. And this is very, much associated with novel species.
Is this what Jerry Berman talks about, jumping genes?
Yes, exactly. Yeah.
And so if you have sweet corn or the type of corn where you have all sorts of different colors of
Yeah.
kernels.
This is very famous example indeed.
And so there is this variability that occurs within the genome that is God's design for
Exactly.
creating immense variety
Yeah.
and beauty. And it's another indication. It's not an indication. It's mistaken for evolution, but it's
But it's pre-specified
right.
variability, and this is all specified in the genome. So, also my final conclusion in my book is also that the genome itself generates the variation.
Mhm.
So the role of natural selection is completely zip, because the genome is making this generation, these novelties themselves, himself.
Yeah, they're describing what is happening. The evolutionists describe what is happening, and they come up with a just so story to explain it. But the just so story doesn't work.
Exactly.
And so we have. Well, one of the things you were talking about was also microRNA.
MicroRNAs, yes.
Tell us about that. What have you found with this?
This is also a very intriguing aspect, because microRNAs are in fact new genes.
Mm-hmm.
Everything in the genome is coded, and what we found over the last, let's say, 15 years, we found a new sort of gene, and and it is an RNA gene, so it means it is not going to be translated into proteins, but it stays an RNA.
Okay.
And we found several of them first about 10 to 15 years ago in a tiny organism, and we thought it is a fluke there, but now we find them everywhere in all organisms. And the interesting part is that all organisms have their own array of microRNA genes. These are orphan genes. If you look, for instance, for microRNAs in chimpanzees, and you look for microRNA in humans, you find more than 2,500 of these genes that are specific for humans. So the whole concept of common descent, of common ancestry, of these two apes, of humans and of apes, falls apart here.
Yeah.
Because you have so much novelties, you have so much novel genes, that if you have to explain this by a natural mechanism, this is really, really not done. You cannot do that.
So this claim that humans and chimps are 98% similar is really a lie, isn't it?
This is really, really not so. This has been known now for a very long time, since 2005, when they sequenced the human genome and compared it to the chimpanzee genome. They already found much more differences. But if you look at the genetic content, this is very, very important, because this is the gene, the tools that specify an organism. If you look at the genetic content, there is at least 10 to 20% differences only when you look at the microRNA genes. So this is incredible.
And what John Sanford showed us at the gene conference was that they were taking the human genome, using it as a scaffold to match the chimp genome, and they would pick all these genetic pieces out and sort of match them forwards or backwards onto that scaffold, and try to make match and come up with this. And then they left out what they called the junk DNA.
Exactly.
And so really there's no such thing as junk DNA, is there?
No such thing. But the interesting part is, if you leave all the sequences out that are different in the two species,
Right.
and you also leave out the point mutations between the
Right.
sequences, then we would be a chimpanzee, isn't it?
That's right, yeah.
Because what they do, they only look at the part of the genome that is already similar.
Mm-hmm.
And they leave out the complete part of the genome that is not present in one of each other genomes. And this is a lot of information. This is at least, as I told you, this is at least 2,500 to 3,000 genes.
So this is a really mechanism they use to construct this lie that we descended from the apes, and and even if you take into account of the fact that the chimp has one extra gene than the chromosome than humans. That even by itself takes the 98% out of it.
Yeah, of course, but this can be explained because what you mean is that chimpanzees have 48 chromosomes and man has 46, and they say that the two of the chromosomes of the chimp have fused together to make chromosome 2.
Right. And
I wrote quite a lot on this topic also. And if you look into it, you can say something for that. But the thing is, if you really look at the fusion point, and geneticist Jeffrey Tompkins looked into this very specifically, and he shows that there is exactly in the fusion point there is an RNA gene exactly in this fusion point. So this makes a fusion in this position very, very unlikely. So probably it is something different.
so these microRNAs, what do? Float by themselves, or are they attached to something, or how do they work?
Yeah, microRNAs are in fact regulators of gene transcriptional networks. You have to look at it like this: genes are never transcribed or never come alone. They work together in networks, and these networks have to be regulated. And it is now like this: you have a few of these microRNAs that can control a whole network because they bind to the RNAs in these networks and then degrade them. And the other thing that I found in my own studies can also be done: microRNAs can also induce genetic expression. So you have you have a bit of both. Mostly they down-regulate the expression of a network. They can also up-regulate the expression of a network. It is all interconnected. It's a living system is a holistic system. You cannot just say there's a gene for this or there's a gene for that. This is really an impossibility. It is a holistic system. It works all together. So also this tells us something about the ideas, for instance, of evolutionists like Richard Dawkins, who is talking about selfish genes, but selfish genes do not exist. Genes work in networks, and as soon as a gene becomes selfish, you get cancer, disease, and whatever.
Yeah, if you want to learn a little bit of nonsense, you just read his books.
Yes, exactly. I fully agree there. Yeah.
Well, I'm really privileged to interview you, Peter. I hope that we'll be at a conference tomorrow, and so it's going to be a privilege to have your conference talk on our available. And we'll see you next time on Revolution Against Evolution. We hope you enjoyed our show.